here to talk to you today about epigenetics. So, I know it sounds kind of scary, but if we break down the word, epi in Greek just means above. So really, the study of epigenetics is just looking at what is above our DNA. So, what the epigenome is it's just a series of chemical tags that are lying above our DNA. So I've got drawn here the chemical structure of DNA. So we have our base pairs, we have adenine and thymine, and cytosine and guanine. So basically, epigenes can modify which genes in your DNA are turned off and on, kind of like a light switch. So one of the ways it does this is a process called DNA methylation, which is when a methyl group bonds to the cytosine pair. So a methyl group would just be a hydrocarbon, which is just CH3. And basically when the body encounters this, when it's transcribing your DNA to make protein, it will come across this methyl group and it'll realize that, okay, this gene is turned off and we're not supposed to transcribe it. So that's one way that the epigenetic modifications can happen. And then another way is something called histone modification. So I got this infographic from the National Institute of Health and I thought it was a really good way to demonstrate how it works, so that's what I'm going to use. So basically, a histone is just this dense packet of chromatin that's solidified into this circle kind of thing. And it works kind of like a spool of thread. So the histone would be the spool and the DNA is the thread that winds around it. So it also has these chemical tails, which right now nothing is attached to because this is like a healthy histone, a normal histone. So whenever the DNA needs to be transcribed, it'll unwind and it can be read by the transcription mechanism. But if we have something that bonds to the chemical tail, like that, then it winds really tightly next to another histone. And the DNA in between these two then cannot be read because they're so tightly wound that nothing can get through to just transcribe it. So that ends up turning off some genes as well, and this would lead to some proteins not being produced. So the effects of this can either be very mild, where you don't have really anything that changes because some proteins we don't need in our body or we only needed when we were growing, like as babies. But sometimes it can have pretty drastic effects. So Feinberg and Vogelstein found that in colorectal cancer patients had different epigenetics tissues than non-colorectal cancer patients. So we found actually in a lot of cancer that epigenetics mechanisms can have a huge impact.